Hello everyone, I've got a KS5 Biology topic here in this video, all about epistasis. Now, I've got a badly drawn, self-drawn chicken over to the right hand side of the screen, and the reason I've included in this video is because that's the first image I show my own students. And I ask them to decide what's the most interesting thing about the chicken. Now, they come up with a variety of answers, but for me, it's this bit here, the comb. Now, whilst there are many different varieties of chicken, it's not just that there are lots of different comb styles because of that. It is in fact to do with the interaction of different genes. Now, in early Mendelian genetics, it was said that different genes or alleles have very specific phenotypes, particular characteristics, and they were unaltered or unaffected by any others. Now, that we've since proved to be incorrect, because what we actually have is an interaction of different gene loci being the location of the gene on the chromosome, so that actually one gene locus can mask or completely suppress the expression of another. And that's what we mean by this title here of epistasis. So we've got the definition here. It is the interaction of different gene loci so that just one gene locus masks or suppresses the expression of another. Now, one of the big consequences of this is a reduction in phenotypic variation. And we really get there's two ways that we can bring about this epistasis. And I'm just going to make a note of these two here. Now, one way is that the genes will work antagonistically against each other, resulting in masking. So they work antagonistically, and that means they go against each other, and that one would result in, as we've said, what's called masking. So you won't essentially notice the effect of one particular gene. So work antagonistically against each other, and that would result in masking. Or the other thing that could happen is that they work in a complementary fashion. Whereby the presence of one gene can help the expression of another. It may be, for example, if you've got a particular pathway where one gene you need to get from A to C. One gene, for example, will help you get from A to B, and the other one will then help you get from B to C. It's where they work complementary to one another. Now, before we come into the different types of epistasis, and I'll talk through one example, I want to talk through two particular words that you will need in this particular topic. One of the key terms is epistatic, and the other term that we need to be aware of is hypostatic. Now, I've put verses there because it really is a toss-up between these two. Epistatic is the gene that is doing the masking, the one that is suppressing, for example, or the one that is leading that. It is the one gene locus that we're referring to in our definition. That is the epistatic one, the one that is prevalent, for example. The hypostatic gene locus is the one that is being suppressed, the one that is being masked. So epistatic is the one that is doing that suppression, for example, if we're thinking about working antagonistically, and hypostatic is the one that is being suppressed. Now, there are three particular types of epistasis. Now, the first of these is called recessive epistasis. So the first one we're going to look at is recessive epistasis. And exam questions love to ask, they'll give a scenario and then they say, can you work out which type of epistasis is shown? So we've got recessive, I'll just list them and then I'll talk through each one. We can have what's called dominant epistasis, and finally we can have what's known as complementary Now, recessive epistasis is where the presence or the homozygous presence of a recessive allele, by homozygous I mean two of the same, homozygous presence of a recessive allele may prevent the expression of another allele at a second locus. So basically having two recessive alleles might actually prevent the expression of another one at another locus, another position on another chromosome even. Now, a good example of this one is the inheritance of flower colour in salvia. Now, whilst I am going to talk about what they mean, I'm just going to include these examples because, again, certain examples like these. So an example of this is the inheritance of flower colour. And I'll give the name of the one that I just gave there in salvia. Now, dominant epistasis is really clear when you've got that. It's where you've got a dominant allele at one gene locus, and the presence of that one dominant allele is going to mask the expression of the alleles at a second gene locus. So simply having one dominant allele is going to suppress or mask the effect of any other particular locus or allele at another locus on either the same or another chromosome. And a good example to think of there are feather colour in chickens. So I'll just put that on there. So feather colour in chickens. And that finally leaves us with complementary epistasis. Now this is where at least one dominant allele for both gene loci present can actually result in them complementing one another. So if one gene codes for an intermediate colourless pigment, for example, the second gene could code for an enzyme that converts the compound into a coloured pigment. So again, at least one dominant allele for both gene loci present can actually result in them complementing one another. And an example of this is crossing white flowered sweet peas to produce purple offspring. So a good example of this is crossing white flowered sweet peas to produce purple offspring. So what I'm going to do now in the video is give an actual example, an exam question, and then I'm going to give you probably the best tip or advice to learn this topic. I'm going to give you the ratios that you will always get in exam questions, and if you just simply memorise these ratios, you'll be able to work out what kind of epistasis you're showing, whether it be recessive, dominant, or complementary. So I'm going to move to a new screen now rather than shrink this one. Particular exam question. I've got two in fact. One at the top, which we're going to try and, using that information, work out what's going on. And then a mini question that I've got that I ask my own students at the bottom, and then they'll need to draw the table, equivalent table of results for this. But I'll show you a quick way to kind of remember this and just to speed you up a little bit. So we've got some data at the top. It says try and work out which type of epistasis is shown here. So clearly we've got some mice. We've got two grey ones mating at the top. And we can see that we're looking at the presence of these two particular alleles. We've got a B and a C, a big B and a little b, a big C and a little c, crossing with an equivalent. And when we look at the sperm and the eggs, we do all the crosses, we've got the outcome that we can see at the bottom. So we've got 9 grey, 3 brown and 4 white. And we've got to try and look at what's going on. Now you can pause the screen at this point and you can have a look yourself, but I'm going to talk through as if you've already kind of spotted that. Now in this example, 2 different alleles are involved. And you might be able to see that the C, the big C or the little c, that one is involved in the presence of pigment. It controls the presence of pigment. And you might be able to spot that the B, so the big B and the little b, that particular allele dictates colour. So whether we're black or brown. Now you might have spotted that the homozygous recessive for c actually means albino, hindering the other alleles from being expressed. So if you have a look here, and I'll just circle these in red, when you've got two of the little c's there, homozygous recessive, and there, and there, and there, notice that it doesn't matter whether we've got a big B or a small b, they're always going to end up this albino colour. It's only when we've got a capital C do we get any form of colour. So we've got a capital C there, we get any form of colour. So what that is implying is that the presence of the two little c's, homozygous recessive nature of the C allele, is actually causing a recessive epistasis. We're hindering the expression of colour there. So this one, you might have got, is an example of what's called recessive epistasis. And that was your answer. Now, all of the following epistatic ratios that I'm going to show you are variations on the normal 9:3:3:1 ratio that we get from a dihybrid inheritance of two unlinked genes. So ultimately, if you just remember this will kind of guide you towards what you're looking for. Notice in this particular question here, we've got a 9:3:4 ratio. If the ratio, I'll put a little title here, ratios. If you see a 9:3:4 ratio, that usually means that we're looking at a recessive epistasis. If the ratio comes out as 12:3:1, or sometimes that can come out as actually 13:1, then you can straight away presume that we have a dominant epistasis. And if the ratio that you find when you do this cross comes out as 9:7, as you might guess, that is when we've got what's called a complementary epistasis. Let's make that a little clearer there. So all you have to do is look at the three ratios you've got there, and it should tell you instantly what kind of epistasis you're dealing with. Now, let's give it another example. We've got this mini question here. So it says that you've got, in corn plants, a dominant allele, capital I, inhibiting kernel colour, while the recessive allele, little I, permits colour when homozygous. Now, at a different gene locus, the dominant gene P causes purple kernel colour, while the homozygous recessive, so little p, causes red. So if plants heterozygous for both alleles are crossed, what will be the phenotypic ratio of the F1 generation? Now straight away from this, we need to look at the parents that we're crossing. And it says we're crossing heterozygous for both alleles. So one parent would be big I, small I, big P, little p. And we're going to cross that with another big I, big P, little p, because they're heterozygous for both, remember? So we're crossing these two. Now, if you were to work that out, if you were to draw a table very similar to the one at the top, what you'd have is something like this. Now, I'll just very quickly shrink the screen here, and we're going to just draw, not the whole thing out. We're not going to draw the whole thing, but just, I'll show you how you can work this out very, very quickly. If we were to do the separate gametes, we would have a big I, big P, big I, little p, little I, big P, and little I, little p. So all the possible combinations. And then if we did this as a Punnett square, again, we'd have the same big I, big P, big I, little p, little I, big P, little I, little p. So if we did this as a Punnett square. Now straight away, we can look at the dominant capital I, because it says that in quantum a dominant allele of I will inhibit kernel colour. So wherever there's a capital I, we're going to inhibit colour completely. So if we were to just put a cross on any particular one where there'd be a capital I, so this I here is the reason why we're putting crosses all down there. Then we've got the capital I at the top here, so we can put crosses here. And then again, with these two rows here, we can put crosses there. Because in all of those, we've got the presence of a dominant, a capital I. So we're going to inhibit kernel colour. Now, if we count up how many crosses we've done, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Straight away, we've got a ratio with 12 in the number. Now, you know one ratio already that exists that has 12 in it, and it's 12 to 3 to 1. So without even doing any more, now clearly in an exam you would need to fill in this table, so you'd fill in these missing gaps. But just to assist you and guide you into how to answer the following questions, we can see straight away that this involves the number 12. This is a 12 to 3 to 1 ratio. So what we're seeing here is a dominant epistasis. Now you could follow this question through and write down the phenotypic ratios of the F1 generation. But again, I just want to really hone in on the idea of working out whether it's recessive dominant or complementary. And very simply, you remember these ratios here and you can't go wrong. Okay, hope all that helps.